And this leads me to say a few words on what I call sexual selection. This depends not on a struggle for existence, but on a struggle between the males for possession of the females. The result is not death to the unsuccessful competitor, but few or no offspring. Sexual selection is, therefore, less rigorous than natural selection.
Now, the basis of sexual selection is natural selection, another Darwinian theory that states that organisms with advantageous alleles and hence advantageous characteristics have a better chance of surviving and therefore reproducing and passing on these advantageous alleles. Now, intersexual selection is concerned with the competition between males within a species.
A prime example is the buck deer or the Cervus alavus. Rival males compete for land and rights to the females located there by locking horns and fighting. Those with larger horns and better fighting technique succeed and hence mate.
Now, intersexual selection involves both of the sexes, and is when one sex, usually the females of the species, choose their partner depending on certain ornaments or displays carried out by the opposite sex.
A fine example of intersexual selection is the peacock. The larger or grander the male's plumage, the more likely it is that the female will choose them, because an impressive tail is a hindrance, so the male displays greater survival ability than its competitors. This suggests they will produce offspring with greater genetic merit. The favourable characteristics do not help the male physically overcome its competitors, unlike red deer.
In male Drosophila, DSX and FRU variants are necessary for development in the P1 region of the brain that is associated with male multimodal sensory processing and courtship behaviour, which is critical for mating success. This suggests that genes in the brain play a role in sexually selective behaviour. In fact, FRU plays a role in wing song, and DSX determines sexually dimorphic traits. The EGR1 gene is a transcription factor involved in social interactions in a number of taxa. For example, in female swordtail fish, EGR1 is upregulated in response to an attractive male. However, it is downregulated in response to other females. There is significant correlation between regulation of this gene and sexual decision-making.
Intersexual selection may also exist in humans as males improve their physical appearance to attract females. Females may choose their preferred male to mate with based on displays, just like in the peacock, as mentioned earlier.
However, over time, intersexual selection has changed due to the advancement of society and the reduction of selective pressures. A male mate may now be chosen because of his wealth or status.
Competition is mainly between males due to the fact that female gametes are much dearer, as there is only one egg released per menstrual cycle and there is a finite amount. Contrastingly, men have a much more efficient method of producing gametes, as they are constantly produced throughout life during spermatogenesis.
A male may be more attracted to females with larger hips, as this suggests fertility and lack of malnourishment. There is less pressure to find a mate entirely based on reproductive and genetic benefits.
These changes in human sexual selection may also be slowing the rate of human evolution, because in the past, favourable characteristics would prevail. However, in the present, weaker humans who would have previously died from selective pressures are more easily kept alive and their genes passed on.